So we have talked about strategies how we can replace cells, but what if we need to do something more complicated, like make an organ? Organs are generally three-dimensional, and you just can't throw cells in a dish and expect them to form these complex and complicated three-dimensional structures. So how do we do that? Let's look at a couple of approaches. So we can use artificial scaffolds or the natural scaffolds, which we will talk about later. But one of the artificial scaffolds which is used is polyglycolic acid, and you can actually make different three-dimensional structures with it. For example, in this case, the researchers are making a urinary bladder. So the strategy is that first of all make a scaffold using something like PGA or polyglycolic acid, the shape of the organ we are trying to manufacture, in this case almost like a sphere, and then you add cells. The cells these researchers used are unipotent cells. Basically, these cells are already differentiated into a specific type of cell. In this case, bladder is pretty much has two major cells. One, the cells which make the lining of the bladder, and also the smooth muscle cells which are present on the outside of the bladder lining cells. So the way these people describe it, it's like baking a cake. So first of all, you make a scaffold of a polymer, PGA in this case. I would like to mention PGA is the same material from which we also make sutures which dissolve on their own. So the scaffold will be chewed up by the cells once they start growing on it. And all we will be left is basically a three-dimensional structure made up of cells and cells only, because the scaffold is going to be taken apart by the cells. So basically we have the scaffold. Initially we add the bladder lining cells, which can be taken from the patient. A small portion of that can be taken from the patients, and these cells can be separated. Remember, we have two types of cells. Using different techniques, we can have the bladder lining cells in one dish, and another dish we can have the smooth muscle cells. So first add the bladder lining cells on the structure. They will start to proliferate, they'll grow. The beauty here is that we don't have to guide these cells like we have to do with stem cells in becoming a specific type of cell. These cells already know what they have to do, and which is that they have to form a lining of the bladder. So they will form the lining of the bladder. After these cells have taken hold, you add the smooth muscle cells, and ultimately, with some more tinkering, you can end up with a bladder in the dish, in your tissue culture dish. So this whole system works pretty beautifully, and these researchers have been able to make urinary bladders, and also they have implanted these bladders into people who have been walking around with these structures, with these organs for more than a decade now. So this was using unipotent cells and also using cells which are unipotent, which are already partially differentiated, if you will. Here's a structure of PGA or polyglycolic acid. I don't want you to memorize it. I just want it to have on the screen so you know what it is. So you can take this material and you can form different types of shapes. Here you can see, for example, it's an ear shape. And what you need to do is now add different type of cells to this scaffold, and these cells can go in the scaffold and start to divide and form the particular three-dimensional structure that you want. You don't always have to use artificial scaffolds. We have abundant supply of natural scaffold, a perfect natural scaffold, which is the amniotic membrane. This experiment was also done in my lab. We isolated amniotic membrane and we cleaned it up. We de-epithelialized it, basically removed all the cells from this membrane. All that was left is basically a sheet of collagen. On this amniotic membrane, prepared amniotic membrane, we added chunks of tissue. This tissue happened to be from limbus. This tissue was placed on this amniotic membrane, and the whole apparatus was inside an incubator with the appropriate media which will promote these cells to grow and proliferate. So these cells started coming out and crawling on to the amniotic membrane, forming a layer. So basically, I should also tell you that limbus is a part in our eye. So people who have corneal abrasion or they have corneal injury, we can basically use this technique to make a cornea. In fact, the cornea was made and was also transplanted into a patient. So this technique works pretty well. We have already, as I mentioned, done this in my lab. We have made these cornea from using limbal cells from same patient. So this was rather simpler organs. What about more complex organs like liver, kidneys, hearts? Not only they have a specific three-dimensional structure, we also have to keep in mind that we have to have vascular supply to these tissues as well. A simple approach, elegant approach could be, for example, we use decellularized organs. So we remove an organ, for example, in this case, liver from a cadaver, and we treat it with gentle detergents, which basically get rid of the cells, and all we have left is basically extracellular matrix or collagen, etc. So here you can see this is a normal liver, and here we have a liver. It is also a liver, but it is basically without any liver cells. And if we inject dyes into something like this, we can see that vascular structure is basically intact. You can see the arteries and the veins. So the vascular structure is intact. So something like this could be used. So we can add different type of cells in this bioartificial tissue and generate a liver. So people are using different techniques with the aim to regenerate lost or damaged tissue. We have talked about some of the strategies. I would like to tell you that this concludes this course, and I wish you best of luck in your careers. Thank you.